It's been amazing to be here since last 2 days. There have been wonderful sessions. First of all, I would like to thank organizers for arranging this conference and having me invited for this one. And special thanks to Dr. Venkatesh. He's been a great support and he's been the main reason I'm here. Okay, can you hear me well now? Is it better? Okay, great. So there have been a lot of talks about breeding and management of chili diseases, various types of pathogen-related diseases. But in my talk, it's going to be slightly different angle. I'm planning to bring in here some sustainable solutions that are derived from seaweed biomass, right? So you're going to hear from me about a new active ingredient from a seaweed biomass that we have demonstrated both scientifically as well as from the field experiments. Before I move into the talk,
E button ಒತ್ತಬೇಡ್ರಿ, E button.
Okay, got it. Yeah, so I just want to have one or two slides introducing ourselves. So we are C6 Energy. We are a proudly Indian biotech company. We started in 2010. It's been about a 15 years journey for us. Mainly, we actually started with the idea of developing seaweed cultivation and using this seaweed biomass for making various value-added products in the areas of agriculture inputs, as well as in animal nutrition and food ingredients, and most demanding area of bioplastic and biofuels. So these are the three different business verticals. Of this agri inputs, we have matured quite well. We have quite a few commercial products, and we have an understanding about technical knowledge about some of these products. So one of that product I'm going to discuss today. So before that, I will. Excuse me. Jumped into the other one. No, I didn't touch that.
This is for the...
Moving forward?
No, sir, this is for...
That's what I am missing. It went far. No, 1 second. Yeah, you can do it yourself. Yeah, you can operate. It'll save time. Go back. No, go to second. Go back.
No, there is nothing, sir.
No, go back. There is a second slide. Yeah, next one. There's a video. Can you play that?
Oh, it's a video.
Next one is a video.
Just a second, I'll play.
Yeah. Okay, so briefly what we do and how we do, I just want to walk you through a video. So this is basically something that we have done for the first time in India, having a scaling up of seaweed production, and
The video needs to have a supporting file also with that.
you don't have.
You should provide it, sir.
Okay, skip it, no problem. Sorry, I think there is some technical issue with the video. I won't be able to play that. Okay, go to the third slide. Yeah. So just coming to directly the topic that I want to cover today. I think yesterday around the same time, Dr. Venkatesh spoke about plant defense activation and how it can be harnessed for plant disease management. So my talk will be in follow-up with that. Especially I will be only focusing on seaweed-based active ingredients. So what we do as a company is we cultivate red seaweed, but in general, if you look at the seaweed, there are 3 types of seaweeds, red, brown, and green seaweeds. So the active ingredients from these have been used from ages. What people have done mainly is to look at the marine algal polysaccharide. So each of these seaweed contain a particular type of polysaccharide, like similar to the land plants which contain cellulose, hemicellulose. These seaweeds contain unique type of an active ingredient. So in case of red seaweed, it is carrageenan. In case of green seaweed, it's alvans, and in case of the brown seaweed, it's alginate. So these alginates have been used quite extensively as a growth promoter and also for abiotic stress management, right? But further, at C6, what we have done is we have optimized the methods, how we can degrade these active ingredients in a selective way to enrich particular functionalities. So in that regard, we have taken this red seaweed, which is cultivated red seaweed, and purified the active ingredients that can do the defense activation. Basically, we have done a controlled hydrolysis process to enrich a particular active ingredient called galactose 4-sulfate. So this is a very unique elicitor. Okay, thank you. This is a very unique elicitor. I will actually show some technical advancement that we have. Can you go to the next slide? Next slide. So this particular active ingredient, we have purified and formulated into a product, namely AG-Fort, which has nearly 80% of this active ingredient, galactose 4-sulfate. And this is a patented formulation. You can see the copy of an Indian and US patent. There are two aspects that I want to cover in my talk further going down. Is one story on how this particular product elicits plant defense, particularly in the case of the tomato leaf curl virus. We used this study in, we conducted this study in collaboration with Professor Vipin at IHBT Palampur. I will go a little deep dive into that concept. And then finally touch upon how much we have tested upon this product in the real field conditions and what is the benefit that we have obtained. Next slide. Next one. So on the mechanism of action, as I said earlier, this was done as a collaboration between Professor Vipin Halan at IHBT. So basically we used tomato leaf curl virus infection model on tomato, and we did the agro infiltration work here. So the plants were sprayed with this product before inoculating, which is an agro infiltration method. And what you see here over various time intervals, 7 days, 14 days, and 21 days, we collected samples and even observations. And as you can see, the treated plant on the right side, the AG-Fort treated, had a mild to moderate symptoms, or in some cases no symptoms, whereas the ones that were untreated had severe treatments, as you can see from the scales and also the leaf pictures that are present on the right panel. On the bottom, what we have noted is the plant height. The plant height is one of the key feature of virus diseases. As you all know, the dwarfing is very significant in case of virus diseases. So you can clearly see the left part, which is control untreated plants, and the right part, the difference is very clear. When we have a AG-Fort treatment, even despite inoculation, agro-infiltration, we clearly see the rescuing of the dwarfing. So further deep dive, we looked at the transcript levels of the virus genes, especially the AV1 and AC1 genes. You can see this is something similar to COVID testing, qPCR studies. So we looked at these genes and clearly understood whenever we have a AG-Fort application, we see a very good difference, very good lowering of these virus loads, especially indicated, if you see that 14-day point, 21-day point, or even 28-day point across both the genes, AV1 and as well as AC1. On the right side, what I have presented is the one of the, the seeing is believing, right? So that is the southern blot picture. You can clearly see the difference, the lowering of intensity of viral DNA in case of AG-Fort treated group, the far right one, compared to the control, which is from a third and fourth from the left one. Next one. Okay, so then further to understand the mechanism of action, we looked at what is causing this kind of a protection in the plants. So we looked at the phytohormone analysis. So basically we applied product and collected leaf tissues and looked at the various phytohormones. Interestingly, what we see is defense-related phytohormones getting endogenously increased. So product actually does not contain any PGPRs or any hormones added. But when we apply the product on the foliage, it clearly upregulates the inside production inside the plants. So you can see the first bar graph showing salicylic acid over 24 and 48 hours, and the second one indicating the jasmonic acid level over 24 and 48 hours. So very clear indication that this product is working through modulation of phytohormones, especially the salicylic acid. Can you go to the next one? Next one. Hello. So then further we did a omics study. So this is transcriptome work. Again, we took healthy tomato plants, applied at that growth stage, foliarly applied product, and then collected the leaf tissue, looked at the transcriptome at 24 and 48 hours time points. This is the data just showing the data quality is reasonably good. Can you go to the next one? Hello. pay attention. Can you go to next? Yeah. So this is just showing the clustering of different genes, upregulated and downregulated genes. So you can clearly see there are certain set of genes which are upregulated, and there are genes that are downregulated. So this is coming out to be a clear gene modifier. So next. So this is just to show you over 24 hours time points, what are the genes that are getting upregulated. So interestingly, what we see is the defense-related hormones, especially the salicylic acid production pathway, and also, very interestingly, for our surprise, what we see is the silencing pathway genes getting upregulated. So go to the next one.
Girish, conclude, please.
Sorry?
Conclude.
Yes, yes. Just 2-3 slides. So this is the data showing, the salicylic acid pathway genes, the PAL5, PAD4, all getting upregulated, and also the silencing pathway genes, RDRs, and as well as dicer genes getting upregulated. So very interesting for us. It was very surprising because in the plant, the virus management pathway is majorly the silencing pathway, and incidentally, this product is impinging on the silencing pathway. So in the next few slides, we were having some validation on how this product, impacting when we did the silencing of, next one. Silencing of ICS1, we show that the salicylic acid production is affected, but in the wild type, it is, AG4 application is increasing it. So go to the next one. And this is again, with ICS1 silencing, we are actually seeing, reduced salicylic acid production. As a result, the ICS1 mutants, we are not able to see the rescuing of the dwarfing. As you can see from the bottom pictures, despite having AG4 application, we don't really see the rescuing of the dwarfness, whereas the wild type 1 is showing the rescuing of the dwarfness. So this is the final model that I have, how this product essentially brings in the changes, both nuclear as well as cytoplasmic, to impress upon the silencing pathway, and thus helps plant to manage against the viruses. Just briefly about 2-3 slides about the field trials, and I'll close the talk. So this is further we validated our mode of action and whatever we see in the lab with the field data. So we did field trials with the foliar applied product in a prophylactic way, and all these trials were done in farmer field at hotspot regions. So this is the data coming from Chilli trials. This is the multi-season data all the way from 2018 to 2021. So basically, across the seasons, we see different levels of disease pressure, but nevertheless, on an average, we see up to 50 to 60% of protection across seasons. And similarly, in tomato as well, here also, multi-season data is generated. Can you go back to the previous slide? Previous one. Previous Chilli, yeah, previous. No, back. One more back. No, you are going front, back.
You need to conclude.
Yes, that's the last slide. I'll touch upon it and conclude. I just wanted to bring up one observation. No, not this one, just back one. That one, that where I had circled that plant. Next one. Next. He's going back. I'm asking him to come front. Next up. After Chilli, there was one Chilli plant I circled. I just want to show that. Yeah, this one. I'll end the presentation with this, Venkatesh. So this is one observation we made in the field. When the plants were early on infected, what we noticed, 2 to 3 application of this product, we are seeing the new flush coming very virus-free. So this is something that, very striking for us. And we usually don't recommend farmers to apply, at this disease pressure, because it's very hard to revert a plant from this kind of a disease pressure. But this is some incidents that we have noted, some observation we have noted from the farmer field. Thank you. So go to the last slide. Yeah. So with that, I will conclude. Go to the last slide. Okay. Go to the last slide, please. Yeah, similarly, we have generated data across multiple crops. I'm happy to talk to you guys, in person. Also, there is a stall, C6 stall. Interested can visit and get more information. In the interest of time, I'm going to stop my talk here with this conclusion that AG4 fits the bill very well in the integrated pest management. If we use all these pest management along with the pest management practices along with the AG4 application, I'm sure farmers will be much more benefited. You can ensure more than 90% protection even against devastating virus diseases. So that's where I'll end. Thank you.